We're going to go ahead for the next speaker, and that is Professor Douglas Weber. Doug is an associate professor in bioengineering department at the University of Pittsburgh, one of the leaders in bioelectronics. He was also a program manager at DARPA and led a lot of the kinds of projects that you've seen over the last several years. So his talk today is going to be on implantable, injectable, and wearable devices for sensing and affecting neurological functions. Please give him a warm welcome.
All right. First, thank you, Raphael and Paul, for the invitation to come visit Rice University. I've been to Houston just a handful of times, but this is my first time spending any significant time on the campus. So I've been a fan of a lot of the work that's been going on here for the past several years. The growth of your neural engineering group is really remarkable. So congratulations on all your past and what will surely be future successes. So my work, even though devices in the title is in the title, my work really focuses on the neurophysiology of neural interface devices. And more, these devices are being developed and approved for use in humans. And the proliferation of both of implantable, injectable, and wearable devices is, I think, really going to change what's possible, not only in bioelectronic medicines, but in many other areas of medicine where interfacing with the nervous system can support unique and powerful functions. So I just need to start with a few disclosures. I do some consulting work on the side, and I get paid for that. So my lab is what used to be just a single investigator lab 13 years ago is now really a vibrant cluster of labs led by independent investigators that each have their own area of expertise, but by working collaboratively allows us to address problems at scale. And that is, we can focus on the many aspects of a problem that limit our ability to implement new solutions, and by working together, tackle it. And I think, as you'll hopefully be convinced through some of the things we're doing in human neural prosthetics, is, I think, really a model of the way this work needs to be done, because these are complicated technologies, complex disease cases, and a single investigator is rarely going to be equipped to tackle all aspects of the problem. So we focus on not so much designing technology, but implementing technology that allows us to both understand as well as restore sensory and motor functions after injury or and also after disease. And so it's basically sensory and motor function that we specialize in studying, and we develop neuroprosthetic devices to interface with different areas in the nervous system to both sense and affect neurological function. And I'll, through the course of my talk, highlight the various devices that we're using for these purposes. This is the group, so there's now nine faculty in total, spread across several schools at the University of Pittsburgh. Several of our faculty are actually clinicians, so I've highlighted in red those members of our group that actually see patients, that is the real doctors on the team, that understand what it's like treating patients on the front lines of medicine, and thus their perspective in shaping the development of new technologies and interventions is really critical. And so many of our faculty have appointments not only at the University of Pittsburgh, but in the medical center affiliated with the university, as well as the Department of Veterans Affairs. We are fortunate to have many active collaborations, not only with other academic groups. I don't see Rice on this list yet, but I'm hoping to add that one before I leave and go back to Pittsburgh. But we work very collaboratively with many other academic groups. As well, we work with many small businesses to develop technology that can be commercialized for eventual human use. I'm a strong proponent of translational research, and I don't believe that you can call yourself a translational researcher if you're not actually working with companies that have devices that are on a road to commercial access. And of course, we're fortunate to be well supported by the various federal funding agencies and private foundations. Okay, so our work focuses primarily on developing science and technology for reducing the burden of physical disability. And I'm showing some statistics here for just a few neurological conditions that result in profound physical disabilities: stroke, spinal cord injury, and amputation. Together account for 1 million new cases of these conditions every year. And for people that have dealt with any of these conditions and know what the current sort of best practices are in this field, you'll appreciate that there's a lot of room for improvement. The technology and the methods that are used to restore function after amputation have certainly gotten better over the years, but they fall far short of restoring what we would consider natural human function. And so our work and my belief is that the only way to really get to the level of function that we consider natural or normal is to connect devices directly with the nervous system. Our sensory and motor systems are responsible for so many aspects of our existence and our interactions with the world that it's only through connections with the nervous system that I think we can provide the level of function that we need. So what do I do for a living? I tap into the nervous system. So I used to work for the DoD. Didn't do any intelligence work. So this isn't wiretapping in that sort of sense. But we are trying to tap into the underlying signaling that is going on in our nervous system when we think, act, and move around our worlds. And so for the people in the room that maybe don't stick wires in the brain, what I'm showing you here are some of the technologies that we use for these purposes. So shown at the center here is an enlarged view of brain tissue showing neurons illuminated with a green fluorescent protein. Shown at the right is a microelectrode array. This is a bundle of small wires that provide several windows into brain function. And by measuring the electrical signals that are generated by these neurons, which comprise our brain, we can examine the pattern of signaling that is occurring in support of many neurological functions. Okay, so if you've never seen the sort of inside of a brain before, and if you've never seen the language of the brain, that's what I'm showing here, is the electrical signals that are generated every time a neuron senses, processes, and transmits information. There's a pattern of spiking activity that is something we can detect with devices like this. Okay, so much of my work focuses on understanding what all of these little squiggles mean, how groups of cells work collectively in networks to support function through signaling cascades like this. Okay, and why does this matter? Because, as I'll show you in the next few slides, we're trying to build devices that use signaling like this to communicate directly with the nervous system. Okay, so we want to enable the brain to have direct control over devices in its environment using capture of signals like this. And as well, we want to be able to go back the other way and restore vision, touch, and proprioception through stimulation of these sensory pathways as well. And again, it's knowing the language of the brain that is going to be key to making that possible. These are some examples of the types of devices that we use. I would say the majority of the work that is happening in the neuroprosthetics space is using implantable devices like this. This is the so-called Utah Electrode array. This is a bundle of up to 100 small needle electrodes that can be implanted in various parts of the cerebral cortex to either sense or stimulate neural activity there. And so shown here is a picture of a human brain with a pair of arrays implanted in sensory cortex. Okay, so by making these electrical connections, we can study population activity in neurons of the brain on a fairly large scale. But those devices require surgery, so we have to open and expose the brain, which requires making a hole in the skull, opening the dura, which is the skin that overlies the surface of the brain. So it's I think Dr. Yosher would say that this is a fairly routine procedure, but for someone coming off the street, they might view this as somewhat invasive. And so less invasive approaches, such as with injectable devices, are also being developed. And so shown here at the top is what's called a Bion muscle stimulator, which was developed now a few decades ago by the Al Mann Foundation. This is a device that can be injected into muscles and nerves of the body and used to provide therapeutic stimulation. I've done work with the Bion in the past, but I'll talk about our work we're doing today with injectable spinal cord stimulators. So now we can place devices like this in the body, target deep neural structures without having to do invasive surgery. Okay, so injectable devices becoming increasingly capable and I think more accessible than most implantable devices. But the ultimate would be something that doesn't require any surgery at all. It would be something that would be as simple as getting up in the morning, throwing on your shirt, and you would have the neurotechnology assistance that you need to support yourself. And so I'll talk about the work that we're doing with a company called Battelle to develop such wearable neural prosthetics. Okay, so just to provide the scientific background, since most of our work really does focus on understanding the language of the nervous system, I like to show videos like this, which really shows kind of the underlying scientific foundation for much of the work that we do. So I'm just going to set this up. This is a picture or a video of a rhesus monkey. He's trained to perform a task in virtual reality. So the monkey's sitting in this chair. He's seeing a screen that is displaying an ellipse. And his job is to trace that ellipse with his left hand. While he's doing that, we're tracking his arm motion with cameras that are pointed at his arm and these reflective markers. And we're measuring through electrodes like this the electrical spiking that is occurring in the neurons of his motor cortex while he draws that ellipse. Okay, and what we're interested in is understanding the relationship between that spiking activity and the movement that's being generated. Okay, so let me play the video. So the popping that you hear, that's an action potential. That's the signal that is generated by an individual neuron. And it's the rate of those action potentials that is the code for the neuron. So what you may have noticed is that there's a very consistent relationship between the motion of the arm and the bursting activity in the neuron. This is a neuron that it tends to prefer or indicate movements that are up and to the left, and so it's groups of neurons like this that form the motor program or the neural code for planning and constructing movements. And so if you can assemble the activity of many such neurons, many such directionally sensitive neurons, we can then decode what it is the motor cortex is trying to do. And if we can do that in real time, then we can use it to control assistive devices. So with that, I'm going to show you a video now of Jan Sherman. So Jan was the first volunteer in Pittsburgh to have these microelectrode arrays implanted in her primary motor cortex. She has a condition called spinal cerebellar degeneration, which has left her paralyzed from the neck down, so she can't move her arms or her legs. Her respiratory function is mostly preserved, and interestingly, her somatosensory function is preserved as well. But in any case, she's completely paralyzed. And so she read about our study online and wanted to be, and literally called us up and said, I want to be the first person in Pittsburgh to do this. So Jan came in. Jen Collinger is leading this project. Jan was implanted with a pair of these 10 by 10 microelectrode arrays in her motor cortex. She had connectors emerging from her scalp for the 3+ years that the electrodes were implanted. She affectionately referred to those as Lewis and Clark because she certainly views herself as an explorer. But over that 3-year period, we were able to study Jan's ability to control a variety of assistive devices using just her brain signals. So now I'll play the video.
Months after the surgery, we came back to see whether she would be able to control the robotic arm with nothing but her thoughts.
So
They
there's Lewis
plugged
and
her
Clark.
brain into the computer, and this is what we saw.
I can move it up and straight down, then left and right and diagonally. I can close it and open it, then I can go forward and back.
That is just the most astounding thing I've ever seen. Can we shake hands?
Sure.
No, really.
Yeah.
Like, come right over here?
Yes, you come over
Okay.
there. Let me grasp your hand there. There we go.
Oh, my goodness. Wow.
And I can do a fist bump if you'd like.
That's amazing.
I think it is really amazing, and you know, I can't emphasize the importance of the underlying neurophysiology, right? It's understanding that neural code that links activity in a group of neurons to some action that the person is performing, or in Jan's case, trying to perform but can't because of her injury. And so it's understanding and being able to model that relationship is what allows people like Jan to control devices like this. The other thing that I think is really amazing is just how arbitrary and free-form her movements are, right? And she's not like intently focused on the arm. She's having this conversation, she's cracking jokes, she's making very impromptu decisions about what she's going to do and what she's going to tell, in this case, Scott Pelley, and it's all very effortless for her. Right, and that's because she's learned how to modulate the activity of just a few hundred neurons in her motor cortex very effectively. Okay, so that's what we're doing. Some of the work we're doing with brain implants. We're also developing interfaces for the peripheral nervous system, including the spinal cord, which is not technically part of the periphery, but by interfacing with spinal cord and peripheral nerves, I think there are many other types of functions that we can support, and again, perhaps through slightly less invasive means. So now I'm going to shift gears a little bit and put my DARPA hat back on. Paul is here, so don't report me to the DARPA police. I spent 4 years at DARPA from 2013 to 2017, and I started a program called Haptics, which was aimed at creating peripheral implants to restore sensory and motor function for amputees. And one thing that was really important for us when we came up with this program is we wanted to have something that was going to be not only high performing in a laboratory environment, but would be robust and reliable enough to take home. Right? We've seen, over the past many years just how exciting and powerful these neural interface technologies can be, but until you can use them at your home, in your community, I think we're, are falling far short of our eventual goal. So I think a hallmark of the Haptics program is that these are technologies that will be suitable and robust enough for home use. So the program involves a variety of implantable devices for measuring nerve and muscle activity that drives movement, and for stimulating sensory pathways in the residual limb to restore sensation. Okay, and so for amputees that have advanced prosthetic hands, the goal is to create a hand that looks, moves, and feels like a natural limb. Okay, so here I'm going to show some work from Greg Clark's lab at the University of Utah. They are working with a number of amputees, upper limb amputees, that have these Utah slant electrode arrays implanted in their peripheral nerves of their arm. So this is an array of 100 electrodes that span several depths of the nerve to record or stimulate activity there. And so I'll just show one of the videos from that. So now, this is a gentleman that actually has, I think, a bilateral amputation. We're measuring, Greg is measuring neural signals from his residual arm, and he's decoding those signals to drive wrist and finger movements in this virtual prosthesis. And you can see that, there's independent control of each of the 5 digits. The wrist has 3 full degrees of freedom that are able to be moved, very accurately, very smoothly. Again, through direct coupling of motor intention in the nerve to movements of the prosthesis. Okay, so that's showing another way to do motor control. Now I want to show you how we're using this technology to restore touch sensation. So using that same peripheral nerve implant, but stimulating rather than sensing activity, this gentleman is going to demonstrate the first time he feels virtual contact through his sensory interface. So just to set this up, he is operating this virtual prosthesis in a virtual reality environment that simulates the contact forces that are generated in that virtual world. So there are virtual sensors on this hand which report when the hand comes in contact with the environment. If we take that virtual sensor data and we send it out to stimulators that are coupled to these electrodes, we can restore that sensation of touch. And you'll see what that feels like for this guy.
Oh my God, I just felt that roar. Oh yeah.
Can you feel the fingers sliding when you do that?
Yeah, actually I can. I can get it on there. God, that is so cool.
But it's so simple, right? It's just it's one feeling of touch sensation. But this is a gentleman that hasn't had either hand. He had both of his hands removed after an unfortunate accident, like 13 years prior to being part of this study. So I think this is the tactile equivalent to going blind. Imagine not being able to feel anything, a loved one's hand, that cold beer that you have at the end of the day. You get so much sort of benefit from being able to explore your world, not just visually and auditorily, but through haptic feedback. And so for this gentleman, having some of that brought back was a pretty big deal. Okay, looks like I better move. At my lab at the University of Pittsburgh, we're doing some similar work, but again, with an eye towards less invasive ways to implement technology like this. We're using spinal cord stimulation, which shown in this animation here. Spinal cord electrodes, such as the one shown here, can be injected through the skin, tunnel down to the spinal cord, targeted laterally where the peripheral nerves enter the spinal cord, and give us access to those primary inputs to the spinal cord. And because those nerves are conveniently laid out almost like the keys on a piano, by taking our electrodes and straddling those keys, providing a number of contacts across a bundle of those nerves, we can kind of provide the punctate sensory inputs to the spinal cord. Again, all in a device that is approved for human use and easy to test. Okay, so our goal is to be able to take touch sensor data from a prosthesis and play it back to the spinal cord as a pattern of electrical stimulation that is distributed across the keys of those nerve roots coming into the spinal cord. And so shown here is some data from 3 different subjects. Much of the work that we're doing right now is really focused on mapping those inputs to the perceptual experiences that the subjects have. So each electrode is overlying a different bundle of nerves, driving sensory inputs that are referred to different parts of the body. And so as we map those keys out, we can see what parts of the body image we can reconstruct. Okay? And as well, one of the things we're interested in is not only where in the limb we can recreate these sensations, but what do those sensations feel like. And so shown in these word clouds are the various verbal descriptions that we've gotten from our subjects describing the sensory experience that is associated with that electrical stimulation. In most cases, there are things that we would associate with as sort of unnatural, electrical, or at least in some cases unpleasant. But as Daniel alluded to earlier in his talk, there's a large parameter space to explore here, and understanding what the right code is for creating pressure and touch sensations is an important part of this research. Okay? But the important thing is that by working with humans with FDA-approved technology, we can make progress much more faster than trying to do a series of animal studies where you can't really have a conversation about what they're experiencing, whereas humans can describe it in often vivid form. One, so the primary goal here is to restore touch sensation such that it improves the amputee's ability to operate their prosthesis. But a secondary, maybe primary benefit in the eyes of the amputee is that by restoring these sensory inputs, their experiences with phantom limb pain seem to be improved. That as many amputees experience what's called phantom limb pain, it's probably associated with the loss of sensory input from the limb. And by bringing that sensory input back, many of these amputees report that their pain goes down. So we think that in addition to the sensory motor functions that this technology provides, that there's an added benefit of pain reduction. Okay, so we're also exploring wearable devices. So we for a number of applications where, especially where permanent restoration of function is not required, if you're interested in therapeutic assistance, then having a device that you can use for a period of time and then eventually grow out of would be desirable. So I'm working with a company called Battelle. They're a research organization, not-for-profit research organization, just a few hours from Pittsburgh. They've developed a technology which we call the sleeve electrode array. This is a garment that has 160 electrodes embedded on its inner lining. When you close that garment up around the forearm, it blankets the entire surface of the forearm from wrist to elbow with 160 of these electrodes, which we can then use for either measuring muscle activity or stimulating muscle activity. And so if we stimulate muscle activity, because we have, again, sort of like the keys of the piano, because we have an array of inputs that we can work with, we can generate a variety of different hand movements by stimulating different electrodes. So if there was wrist movement, we had different patterns of finger movement and so on. And again, because these are like the keys of a piano, we can construct arbitrary symphonies with input across that array. Okay? And so the goal is to create a fully wearable system such that there's a portable box that controls stimulation through the sleeve. It can be worn on a belt and then used in the home to support activities of daily living. So I'm just going to play a video here showing a volunteer at Battelle in Ohio State that has C5, C6 spinal cord injury, and so he has very limited hand function, but using stimulation delivered through this sleeve, he's able to grasp, in that case, a tennis ball. Okay, so this is using a portable array, portable and wearable technology for reanimating hand function. As I mentioned, though, we can also use this as a sensor array, right? So in addition to being able to stimulate muscles that are paralyzed, we can also measure the activity in muscles, which you might think, is there any electrical activity left in the muscle if the hand is truly paralyzed? And the answer is yes, and I'll show you how important that is in a few slides. But now we have a way to both sense what's going on in the arm. We can use that information for diagnostic purposes. We can identify unhealthy patterns of coordination in the hand after stroke and spinal cord injury. And then we can use electrical stimulation to treat deficits or to restore healthy coordination of muscle activity, all in a single garment. Okay, so I'm going to show a video now showing our ability to decode hand gestures from that pattern of muscle activity. So shown at the bottom left of this screen here is an avatar which is displaying a target gesture, and then the center avatar shows the decoded gesture from the muscle activity. So by looking at the pattern of muscle activity that is generated during these different movements, we can predict with very high accuracy what's going on in the hand. Okay, and so if we can do that in people with impaired hand function, this gives us a way to control assistive devices in a very non-invasive wearable form. Okay, so here is just some very early work showing what we're doing in stroke. So here we have a person with very weak hand function. His ability to extend his wrist and open his hand is highly impaired because of his stroke. But if we look at the underlying patterns of muscle activity that are generated in the impaired forearm, we see that even though he's attempting but able to perform only weak movements, there's very strong, easily detectable activity that we can use to predict the different classes of movement that we're asking him to perform. Okay, so this is a very impaired hand that still reflects, I think, a very strong will to move. Okay, so that's after stroke with some preserved hand function. This is work that we've done at the University of Pittsburgh with one of our BCI participants who has absolutely no hand function. Okay, so because of his cervical spinal cord injury, he can't move any of the digits of his fingers. Yet when we ask him to try moving those digits, we're able to detect very strong bursts of muscle activity in his forearm. Okay, so that means that there's spared connections between his brain and his muscle. There aren't enough to generate functional movement, but there are enough to be able to be detectable using our sensor array like this. And if we look across a range of individual finger movements, we can decode with very high accuracy which movement Nathan is trying to express. Okay, so that means that there's sufficient information in that impaired forearm to drive assistive devices. Okay, and it looks like I'm out of time, so I'm just going to wrap up by saying briefly that more and more we're trying to shift this technology from in-lab use to home use, and creating wearable and particularly portable systems, not just individual devices, but whole systems is going to be essential for that. And I hope I've convinced you that by coupling technology directly to the nervous system, there's a wide variety of, I think, very useful functions that we can support. And more, these advanced capabilities are becoming accessible for human use because of advances in injectable and wearable devices. And so with that, I'll take any questions.
Excellent. Thank you, Doug. So if you have any questions, line up at the mic. We also have some good questions online, so if you still want to put in some questions online, go ahead and do that. You have a question? Go ahead.
What controls the dexterity of the prosthetic? Is it the spatial resolution of the electrodes?
For the BCI work?
Correct.
Is that what you're talking about?
Yes.
So that is a function of the decoding setup that is implemented. So that arm that you showed, that I showed in the first video, that has 22 degrees of freedom, but we don't use all 22 degrees of freedom in all cases. So we start with a simple task, 2D movements in a plane, then we move to 3D, eventually 7D, so you have reach, orientation, and grasp. And they've gone as high as 10D, I think.
Perfect. Thank you.
Yeah, sure.
I thought it was very interesting to see that even in spinal cord injury cases that there were still connections going to the muscles. I'm curious, is that something that's generally clinically assessed? Is there a widespread appreciation of whether those signals are there for a lot of people, or was this an exception?
So other groups have used muscle activity below the level of the lesion for control like this. Generally not to this level of dexterity, though. I mean, we're decoding individual finger movements. Usually it's just hand open, close. But I think more and more the field is appreciating that just how much spared function there is, even in people that are diagnosed with complete spinal cord injury. There's been a lot of work in the last year showing publications of recovery of voluntary function after spinal cord stimulation in people with paraplegia and clinically complete, motor complete spinal cord injury, right? So if there is any recovery of voluntary function, that means that even though the clinical diagnosis is complete injury, that means that there's some spared pathways that are again, too weak to show function, but maybe suitable to support a neuroprosthesis.
Would you have a recommendation to neurologists or rehab specialists, could there be this population of people who could be immediately helped?
Well, so I think what we need are better tools for diagnosing or predicting who might be candidates for some of these advanced technologies. And I think one of the one of our goals for this sleeve array technology is to obtain a better estimate of the extent of spared connections that are there. So we just started a project with a group that has expertise in motor unit number estimation, so they can count like individual motor units in an electromyogram. And so if we can actually put a number on the number of spared motor neurons that are there, then that might be useful for predicting, this is a person that is a candidate for occupational therapy and recovery of some natural function versus, this is someone that's going to require an assistive device.
Yeah, that's really cool. Thank you.
Sure.
Let's take another one over here.
Hi, for the implantable devices, how important is it to target the specific neurons that before the damage would have controlled that effector versus could somebody just be retraining themselves to use a different neural population
Yeah,
to
so
control
that's a really interesting question. My personal belief is that you should try to get as close to that original structure as possible. You can teach an old dog new tricks. You can learn new skills, but it's just harder, right? And so the closer you get to the, to the original area responsible for that function, I think the better you'll be. So, it's something we're looking at on the sensory side as well, because we would like to be able to remap sensory areas, because it's actually a little bit more difficult to map out the sensory parts of the nervous system before placing these electrodes. And so if we could shift receptive fields or projected fields post hoc, that would be very beneficial.
Okay, one more.
As opposed to patients or subjects who have undergone amputations or paralysis, who have used limbs before, have you ever tested these technologies on people who are born with inabilities,
Congenital,
or disabilities, or
yeah.
who had disabilities such at young age that they never used arms or legs before?
So I'm not aware of any studies in the somatosensory system that have looked at that. For sure there are people with congenital defects, and missing limbs, that are born that way, and they are able to learn how to use myoelectric prostheses. There hasn't been any work, as far as I'm aware of, on the somatosensory side, but in the auditory field, cochlear implants, are given to children that are born deaf, and they're able to develop functional hearing, speech perception, using, with training with the cochlear implant. So I think the brain has the capacity to, to take on those skills, even if it, maybe didn't start that way.
Great. Okay, so we have one Slido question. So how long does it take to train a patient to control a prosthetic device?
So at that point in the study that we showed there, Jan was approximately a year into the study, but literally on the first day of testing in the lab, she was able to do 2D movements. And she probably could have done 3D movements. We just didn't do 3D testing that day. So, I mean, it's super fast because, we try to approximate as closely as possible the natural coding scheme for the neurons. Right. Okay, and the way we sort of unmask that in someone that's paralyzed is we show them videos of the movement that we want them to generate, and through what some call the mirror neuron system, it generates observation-related activity. So as you're watching me wave my hands, your motor cortex is lighting up and encoding hand movements. Okay, so we can evoke through a visual display the natural coding scheme that we want Jan to use, Jan and the others to use in driving the prosthesis. And that works very well. And so,
so it's very intuitive.
It's very, very intuitive. And then that's really important for the learning aspect of it, right? If you're going to ask somebody to learn a complex skill, then their ability to translate feedback into change in function is really key.
Right. Excellent.
Caleb.
So the video that you showed when you were talking about somatosensation was very interesting because the student asked the patient to slide. Sliding or texture seems like it's one of the most complicated features to stimulate because it involves both spatial and temporal.
No doubt.
So it's related also to the end stuff. To what extent do you think that it's critical to be able to control, to do sort of simultaneous activation versus sort of temporal pattern? Like, are they both critical, or is one more important?
Can you repeat that and just
Sure.
so
So,
everybody
so the question is, what are the essential, correct me if I'm wrong, but I think your question is, what are the essential features of the sensory code for something like touch or texture sensation?
But presumably the same question applies also in the effector space.
Sure. Well, I think we know a lot more in the effector space, but your question was on the sensory side. You know, the reality is we're still at such an early stage, and we're at least 10 years behind the motor folks, right? Because the motor work has been ongoing since the early 2000s in humans, and it's really just in the last 5 or so years that we're doing work like this in humans on an appreciable scale. So, you know, so we're still very much working at a single or small channel count scale. So addressing questions related to the spatial patterning of activity, I think we're not ready to do. On the temporal side, one of the challenges is, you know, so the sensory nervous system and probably many neurons in the nervous system work this way, but undergo habituation or adaptation. You know, so when you put your shirt on in the morning, you feel it for a period of time, but then eventually you ignore it because your brain adapts and it filters out what information which it presumes to be unimportant. And so if you bombard the nervous system with electrical stimulation like this, the sensation will be strong initially, but then attenuate gradually. And so figuring out how to work with that adaptive encoding scheme in the nervous system is one of the many challenges that we're facing.
Okay. All right. Thank you again, Doug.
Yeah.